This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 298, recorded on October 26, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone.
From Ann Arbor, Michigan, Michele Swanson.
Hello, good to be with you.
And from St. Louis, Missouri, not Washington, St. Louis, Missouri, Petra Levin.
It's good to be here.
I get tripped up on where you are. I don't know why.
I know. They had to change the name of the university to make sure people weren't confused in the 70s to Washington University in St. Louis. That is our official name.
Oh, that's the full name now, really?
Yeah. Well, it's been that way since the 70s, apparently. But I still get asked about Seattle weather.
You know, St. Louis also throws me because when I see Louis, sometimes I say Louis, even though it's spelled differently. Louis
we're
Armstrong.
named after, a Louis.
Yeah.
Spelled Louis.
St. Louis, Missouri. It just doesn't slide off the tongue like everything else. Oh, well, I have to get used to it. Anyway, welcome back to TWiM, where we try to teach you microbiology. And if you enjoy it, if you like our teachings and our method, which is to have discussions, we'd love to have your financial support. You can go to microbe.tv/contribute. Microbe.tv is a nonprofit corporation, which means that your contributions, as small as they may be, are still federal U.S. tax deductible. So, if you give $1 a month, $12 a year, you can deduct from your taxes.
Only if you itemize.
That's true. And I never reach the level of itemization. It always says you're going to get the standard deduction. Sorry, ever since, I don't know, one of the presidents changed the tax law, I used to deduct my real estate tax and all that. And nope, no more. It's all done. Got to pay more money. Anyway, but we'd love your support, irrespective of whether you itemize or not. Microbe.tv/contribute. Today, some very, very unusual topics for you. And Michael's going to start us off with a snippet.
Well, as I was looking for papers this week, I ran across this editorial from Nature Microbiology entitled Better Training for Non-Academic Careers. And I guess I was biased because we had a meeting with our vice president for research earlier that week, and she was talking about the need for us to recruit more graduate students to help the faculty with their research. And the central theme of this editorial is that as more PhD students and postdoctoral fellows transition to professions outside academia, training programs must prepare scientists for these alternative careers. And so as I read that central theme and this single one-page editorial from Nature Microbiology, I thought it would be something that we should at least have a conversation about because many of our early research career scientists are struggling with this. Traditionally, the career path that I myself took is I completed my bachelor's degree, and then I went on and obtained a master's degree before moving on to a PhD. I then did, like many of us do, I did several years of postdoctoral training where I had an opportunity to do intensive research, learn some new techniques, and I had an opportunity to teach while as a graduate student as well as when I was a postdoc. And then I went out looking for that ideal tenure-track assistant professorship at one of our institutions of higher ed. And I was lucky. I was one of the fortunate ones that was able to find a job where I had high job satisfaction and was able to progress. But that's not always the case for many of our PhD trainees who do successfully complete their degrees and move on. Now, this article or this editorial quotes Dr. Pat Schloss, a colleague of Michele's at Michigan, and he authored an opinion piece in 2018 that appeared in MSphere. And recall that MSphere is an open-sourced journal, so you can read Pat's thoughts on this. And it was entitled "In Defense of an Academic Career." And I'm going to quote Pat for our audience who may not be aware of the trials and tribulations that all of us face in academia on a day-to-day basis. And here, quoting Dr. Schloss, about 10% of the proposals that I have written have been accepted for funding. I submit manuscripts expecting them to be rejected. I spend half my work hours either answering emails or in meetings. I've been negotiating access to a set of samples for the past 5 years with no end in sight. If I want to use an autoclave, I have to take a 3-hour training module. I struggle to get a transparent report of the burn down rate on my research budgets, and my value to the university appears to be linked to the amount of funding that I bring the university. Although a paper of mine may be cited dozens of times, it's discounted because it is published in a journal with a low impact factor. My most impactful piece of work is a software package that is literally used by thousands, and yet it has not had direct funding for the past 5 years. In the same year that I was promoted to full professor, an indication that I have an international reputation in my field, my department told me that I needed to cultivate an international reputation. Despite several high-impact findings, I cannot pique the interests of our intellectual property or development offers. So Pat then goes on to say, why do we stay in academia if we are so miserable in the ivory tower? And here's the real truth why we all stay in academia.
And here's a big pivot coming up.
Yeah, because the truth is, despite all those pinpricks and our moaning, we have some pretty damn good jobs. So Pat goes on to highlight in his opinion piece the positives, which include academic freedom, leadership opportunities. And I think the most inspiring thing I find in my job is working with our talented trainees and networking with people like Vincent, Michele, and Petra. And it's that working with others that actually keep you intellectually stimulated and provides that job satisfaction. However, in their editorial, Nature Microbiology offers that the Royal Society in 2010 offered that only 0.42% of the students earning a PhD will secure a professorship at a university. And I found that stat to be really pretty frightening, especially if I was...
Does that specifically refer to in England?
That I didn't dig
the United
into.
Kingdom?
And what is the PhD in?
It doesn't specify,
because
but
some PhDs not designed to be in academia at all.
And that's one of the things that I think we in the sciences need to drill down on and begin to think about. And the Nature editorial goes on to say, of course, this varies by discipline and country, and there may be alternative career paths within the sphere of academia. It is being increasingly acknowledged that there is an imbalance between the number of early career researchers and the number of available faculty positions. And I think the other reason I gravitated towards this is I had just read a piece in the Chronicle of Higher Education where we watched with concern as West Virginia University is going to lose 28 academic programs and 143 faculty jobs from its flagship Morgantown campus following a vote from its Board of Governors. And recall that West Virginia University is a very modern, Research One land grant university. An R1 institution is seeing the contraction. And granted, those 28 programs aren't in engineering or the hard sciences, but nevertheless, it really speaks volumes about this issue. And maybe we need to begin to think about helping our trainees recognize that they need to have a little bit more breadth in their training in case they don't get the job satisfaction that the four of us, well, I shouldn't speak for the others, but at least me, has experienced in our collective careers. And so this article is chock-a-block full of good advice for those people who are thinking about getting a PhD and what they need to be doing while they're going through things. And this brings me to my first question for our group here this afternoon. What would you imagine would be the requirements for our graduate training programs to facilitate the preparation for non-academic jobs?
So I find this very dated, this idea, because it's been at least a decade since my graduate program, at least, has been thinking about this idea. So, yes,
this is what I was,
I'm just
scratching
kind of
my
surprised
head.
that someone's writing this in 2023 when really people were thinking about this in 2010.
Yes.
Doing more than
generally,
thinking.
Yeah, I mean, we've put in a lot of things. Hopkins has, yeah. So that was my first comment. But the other thing that I find odd and that maybe I think we should discuss is, when we talk to our students about what the purpose of a PhD is, we get very different answers because I think there's confusion about why you're getting a PhD. Whereas if you ask somebody why they're going to med school, they almost always have a very straight up answer. So
yes.
I think that needs to be clear that it's a professional degree.
And the exciting news is that there are a wealth of careers that are really impactful and advance society and the public, the greater good, that require a PhD, but are not doing bench work and training 5 more PhDs. So
Right.
I think not PIs, because what we know as faculty is we know how to be good faculty and productive faculty members. We need to set up at each institution and in our professional societies career and professional development programming and encourage our trainees to explore the wealth of opportunities out there, invite back our alumni who are now thriving in careers outside the traditional academic track, and provide skill building so that people can explore and be competitive for the job of their dreams. So here at Michigan, about 30% of the PhDs end up in an academic career. But guess what? The unemployment rate for the other 70% is not 100%. I mean, they are thriving in a wealth of careers. Genentech, for example, was not even around, I'm sure, when a number of us were training. So, I mean, there's a lot of excitement in our field, and I think we need to get that message across and deal with the conflict of interest that some faculty members want their trainees to be at the bench 100% of their time, and it's not practical. We have to open up and broaden what training is, and it includes exploring career paths and getting other skills that are transferable.
I've been at programs where they have regular seminars for the students, people with different careers who come in and talk about them, right? Not just the usual data talk, but someone comes in, I work here, I work at this kind of company or that, and I use my PhD in this way. And because students have no way of gathering that kind of information. It's really their department's job to do that. But as you just said, they all want you to just do research and not do anything else. Right?
I think there's just a conflict because I think the university, or at least the graduate programs, that we have a big umbrella here at WashU, but they recognize that this is, that academia is the minority of our students will go at least into academia as their final, after doing a postdoc. And so they do have a lot of programming, but I think, there's still a lot of confusion among the students and faculty. And I think all the things that if we basically sell a PhD for what I think it should be, which is you're being trained to study a problem deeply, to manage a problem, to solve a problem, to, think about, what the endpoint is and to communicate your data in a clear way.
Work in a team.
Work in a team.
All
absolutely.
of those that's what you're learning in a PhD, and that's applicable for so many things. But if you think a PhD is like, I'm going to come in and study something really deeply, and that's the whole point, then I think you're missing the point. And I think we're not being clear about what this really is. And if faculty are confused that, the students are really there as
to make them famous.
To make them famous.
Their faculty mentor famous.
They probably shouldn't be mentoring trainees. They should be mentoring technicians. And, how do we essentially bring everybody along to this idea? Like, we're a professional degree, like a medical degree or a business degree, because really in the end, we might use a different tool to get there, but that's how I see it. And I think if we see it that way, then we'll be training our students the right way.
And so what I've done is I put into the show notes a number of links. First, to the references that they mentioned in the Nature Microbiology editorial, but more importantly, for those of you in the United States, the American Society for Microbiology, our sponsor, has a career site that is chock-a-block full of good things. And so if you're struggling to figure out your next evolution or where you're going to go, or you're just a brand new graduate student trying to navigate this complex path, there are many resources available, and I try to identify them for you because you're our future. You are going to provide the insight and the breakthroughs that we so desperately need in science today. And as Michele articulated, there are many career paths where a PhD is not necessarily training the next generation, but making those strides in protecting society and providing public health services and doing many of the things that we candidly all take for granted.
And driving translational science.
Absolutely.
The article does link to the Nature Careers Toolkit, which has terrific resources for people to, at no cost, explore different avenues and how to learn more about them and make steps in those directions. And they also include links to another website, Versatile PhD, which is for people that have PhDs not only in the biomedical sciences, but social sciences as well, and then a few other resources. And I'll also add both the National Academies of Sciences and the National Institutes of Health are currently have working groups diving deep into postdoctoral training and what do we need to do to make it a better fit for the current opportunities that are out there.
So I thought this would be a good discussion. That's all I had planned on bringing to the table of this, is to really say that we're not late to the table here. This isn't a cutting edge editorial. As Petra said, this has been an active discussion and active implementation, not only here, but from what I read in the UK, they have been working this problem for quite some time as well.
And I'd encourage those junior scientists who are considering a PhD program and looking at different programs, one question you can ask is, tell me, where have your trainees gone, once they finish their degree? What are they doing now? And if they don't have a robust pipeline to a wealth of career options, then you want to wonder why that is. And why don't they have a career and professional development team that is providing those resources and panel discussions, et cetera?
And I think it's important for students to even ask those questions if they think they want to go into academia. They should be asking those questions because, when I was 22, I'm pretty sure I wasn't exactly sure what I wanted to do. And I kind of went into a PhD with that idea. But to know, as you progress, maybe your idea will change. And so if those resources are important no matter what you think are important when you first start grad school, you want to make sure that they're in place in case you do change your mind.
I'll also add that I'm sure each of us would agree that if we look at our own portfolios, every 5 or 10 years, it changes dramatically. So even though our job description, academic scientist, has not changed, how we're spending our time and the efforts we're contributing to changed quite a bit. And that's a wonderful advantage of academia. We get a lot of choice in where to put our efforts, and they're great opportunities. And podcasting is one of them.
Well, let's not forget Vincent's evolution. He evolved from an endowed chair at Columbia University into the, what is it that Martin refers to as the Podfather?
Yeah, the Podfather. I love that.
Podfather. Yeah. Well, that's the advantage of tenure, right? You can try different things. It doesn't mean you're going to keep your grants, because that's right. That needs an active lab. But if you're tenured, you have a job and you get some salary and you can try different things. Although I started podcasting long before that, and I always wanted a second career, and so I just took advantage of tenure and pursued it. And I think for young scientists, you should do some kind of communication for sure. But your primary goal is to do either research or whatever else you're doing in your job. But we all need to communicate more, and I'm trying to set an example. You have all these wonderful people who do it with us.
As another example, for more than 10 years, I've been directing the postdoc office at Michigan Medicine. So we have more than 600 postdocs who come from all over the world, and we work on setting up equitable policies and also programming so that they can make the most of the resources at University of Michigan and launch the career of their dreams. So that's been very satisfying as well.
So I think also good grad programs that have been thinking about these alternatives, they often have in place. And a lot of these are student initiated, but groups that essentially allow students to explore different kind of careers. I mean, at WashU, which I don't think is that unusual, we have ones for consulting where you can actually do free consulting for local small businesses in biotech. We have one for policy, and we have one for teaching, and we have one for communication and editing. So I think this is pretty standard at most places. And if you're interested in exploring these kinds of groups, are excellent, and my students have gotten a lot out of them. And so I encourage any grad students out there, new students or students thinking of graduate school, to look and see what's available at your university if you want to try something.
You not only learn more about particular fields, but you also then can network with former graduates who are in that field, and it's a great way to explore.
And I thought that I selected this paper because we're now approaching the graduate school application cycle. And so this is a good podcast to listen to, so that as you're picking the schools that you're going to either go and visit or perhaps apply to, these are some of the questions and programs you need to be looking for to make certain that you get your best experience possible in the time that you're going to invest in whatever degree you happen to be pursuing, whether it's a master's, a PhD, or even an MD or DDS.
All right. Thank you, Michael.
Thank you.
And Michele, what do you have for us?
I have a paper titled Airway Microbiota and Immune Mediator Relationships Differ in Obesity and Asthma. And this was published in the Journal of Allergy and Clinical Immunology in April of this year. And the authors are Ari Kozik, Lisa Begley, Nirja Lagogo, Alan Baptiste, John Herb Downward, Christopher Oprin, and Yvonne Huang. And they are from the University of Michigan in the Department of Internal Medicine's Division of Pulmonary and Critical Care Medicine and Division of Allergy and Clinical Immunology, and the Department of Microbiology and Immunology. So let's start with obesity, the main focus. So according to the CDC data from 2017 to 2020, 42% of the U.S. population is obese. And this is defined by a body mass index greater than 30, which if you're 5'9" tall, that means you weigh more than 200 pounds. So just to give you a feel for that. And people with obesity are at increased risk of heart disease, stroke, type 2 diabetes, and some cancers. And it's estimated to result in an increased annual medical costs in this country of $173 billion. Unfortunately, people with obesity also have a harder time managing asthma symptoms, and they seem to be at a higher risk for severe outcomes. And they They don't present with the typical clinical profiles of someone with asthma or the typical disease course or response to treatments. And the first author, Ari Kozik, also points out that people with obesity often struggle to receive equitable care due to stigma. For example, their symptoms might be dismissed, they might not be taken seriously, they might be told just to lose weight. So studies and discussions that focus on people with obesity, people with asthma, need to be designed and analyzed thoughtfully. So now let's consider asthma. This is a chronic inflammatory disease that is marked by episodes of wheezing, breathlessness, chest tightness, and coughing at nighttime or in the early morning. And according to, again, the CDC's National Health Interview Survey over a 8-year period, in the U.S., about 8% of the population suffers from asthma, but it's not even across the population. Emergency department visits are more prevalent among children, women older than 18, and all racial and ethnic groups. Asthma is also more prevalent in low-income households, and it's more prevalent in small and medium metropolitan areas than in large central metropolitan areas, etc. Most disturbing, Black children are 2 to 4 times more likely than white children to be hospitalized or die from asthma. The first author, Ari Kozik, actually published a commentary in M-Sphere, the journal that Michael referred to, in 2020, and it's a position piece titled Frame Shift, A Vision for Human Microbiome Research. And it is basically looking at how we need to think not just about simply as race or ethnic disparities, but address the larger problem. So she specifically discusses 2 pieces, one entitled Time's Up to Adopt a Biopsychosocial Model to Address Racial and Ethnic Disparities in Asthma Outcomes by Matsui et al. In 2018 Journal of Allergy and Clinical Immunology, and another piece by Finley et al. In Trends in Microbiology of 2016 called Health Disparities and the Microbiome. And Dr. Kozik points out in her piece that traditionally studies that invoke the Black body as a biological explanation for disproportionate disease burden in the U.S. are overlooking many other important factors. And Ari describes her own childhood memories of an ever-present smell and thick, hazy blanket from steel mills, oil refineries, lead smelters, and chemical plants, all located within a 10-mile radius of her elementary school in northwestern Indiana. And that county was 95% non-white, and it also had the state's highest prevalence of asthma hospitalizations. So she makes the case, as do these authors that she quotes, that we got to move beyond race. It's too often used as a convenient measurable proxy, but it's really a social construct that doesn't take into account other critical factors such as environmental stresses, social stresses, access to healthcare knowledge, and providers. So they advocate for a biopsychosocial model when we do microbiome research and health studies that to take not just what race are you, but rather looking at physical environment, economic and psychological stress, and access to quality health care. So I hats off to Ari, who's been a real advocate for the Black community, and she is applying that in her research that we'll talk about today. So they're interested in what features might contribute to the observations that people who are obese and have asthma present differently and they respond differently to treatment. So in Dr. Huang's group at University of Michigan, they've been able to leverage 2 ongoing clinical adult asthma studies where patients with mild to moderate asthma come in voluntarily to clinic for a variety of clinical measurements, sample collections, and monitoring. So this is a prospective observational clinical study that they've been able to tap. And we'll talk about that in detail. But initially they were motivated because when they looked at the microbiome data from these ongoing clinical trials, they noticed some clear differences between people that were obese and people that weren't. And they also knew from the literature on the gut that the microbiome profile can affect risk of inflammation and certain diseases. So they set out then to take a very systematic approach and apply their computational toolkit to look at associations unbiased between particular clinical traits, cytokine profiles, and the microbiome composition. So in particular, they wanted to know, is obesity associated with alterations in the microbiome of the airway of people with or without severe asthma? And are there particular interactions between certain immune molecules, cytokines, with the airway microbiota that would distinguish adults with asthma who are obese or not obese? So this is obviously a very ambitious and complex issue, but they have got the skills to really build a foundation. And let me also just say by way of introduction that even the study of the lung microbiome field is relatively new. Certainly when I was being trained, I was taught that the lung is basically sterile. My bone marrow or my lung macrophages are eating anything that comes in sight. But in the last 10 to 15 years, now that we have many culture-independent methods, DNA-based methods, to really interrogate the lung and look at what is there, we find that, yes, it too has a microbiome, which of course makes sense. We're inhaling air that's full of spores, fungal spores, microbes, we're eating microbes. Of course, our lung is going to have some. So again, their goal is now to really look at the associations between what's in the lung, what microbes are there, and then compare that to the clinical profile and also cytokine levels and even the gene composition of the microbes that are there using their really impressive toolkit of computational methods. So their ambitious plan is to obtain blood and sputum from these generous patients that come in for their clinical studies and get cytokine profiles using multiplex assays that involve antibodies specific for different components of the immune system. And then they also collect from each patient that comes in a sample of their airway microbiome. Now, you can imagine that would be very challenging, but the field has determined that you can get what's called an induced sputum sample, in which a patient will first inhale over a period of 12 minutes saline, and by inhaling that moist saline, it liquefies the airway secretions, causes coughing, and actually expulsion of secretions from down in your bronchial cavities. So previous work has demonstrated that this induced sputum technique, you are able to obtain samples that have a similar microbiome composition as brushings from the bronchial airway. And that just sounds creepy to me. I can't imagine anybody.
It sounds less invasive than a bronchial alveolar lavage.
Yeah. And do you want to tell us what that is, Michael?
They literally drown you, I think, is they pour liquid on top of your lungs to facilitate the expelling of the airway contents.
And is a person sedated for that?
No.
They're not.
Oh, it just sounds miserable.
Sputum for the win, I think.
It's not. Yes. It's not a great procedure. Yeah.
No, it's not good. And so I like the moist saline to stimulate the coughing and liquefying because it makes more sense that it's sloughing things off, and you'll have a representative sample of who's likely to be living down there. Yeah. Because that's what you're trying to see.
That's right. And of course, these are still much more precious samples than, let's say, going to the warm room and pulling out our culture of, your favorite microbe.
Or a stool sample,
That's
which
right.
people are gladly to get rid of.
Yeah. So Ari, our first author, did want to give a shout out to their co-author, Lisa Begley, who was essential in the painstaking specimen processing pipeline that they needed to then generate high quality DNA sequence data, which allowed them to then profile who's there, which bacterial species are there, which fungal species are there. And of course, to do this required a lot of computational analysis, and she wants to highlight her colleagues and co-authors Chris Operon and John Herb Downward, who were the data wizards and helped develop and testing the methods and troubleshooting the methods. So tables 1 through 3 outline the patients that generously contributed samples that they were able to bank and use for this study. And they do a good job of describing what the clinical features, what their weight, what their age, their race, et cetera. And also they describe in detail in the methods section a number of clinical tests they do to actually quantify what the respiratory capacity is of the people and other kind of clinical hallmark traits that I won't go into. But it's really a very complete analysis of profiling the patient population that they're getting these samples from so that they can later go back and ask particular questions and have a better understanding of what their denominator is when they're comparing different groups. So the first question they asked is the bacterial burden similar in people who are obese or non-obese? And they found, yep, they were pretty similar, no dramatic differences in the overall burden. But when they did the genome sequence analysis, they found that the community composition actually differed between people who were obese and those who were not obese. And that data is shown in figures 1b and figures 2b. Figure 1b, for example, is a nice color-coded bar graph. And we see that in the obese patients, they've got a Gemella species, a Prevotella, a particular Streptococcus, and a few others that are prevalent. And those are not common in the non-obese sputum bronchial samples. They then want to, having obtained information about the microbial community, they wanted to gain insight into the inflammatory status. So again, they had this sample from the lung, but also from the blood and from the blood in the sputum, and they used their cytokine profiling to develop heat maps to look at what cytokine and immune mediators were present. And that data, and there's a load of it, is summarized in some really easy to assimilate heat maps shown in figure 3. So we look at 4 different quadrants, and you don't have to know anything about the biology or the computational methods. If you just look at these 4 quadrants, you can see quite clearly that people who are not obese and have asthma or don't are fairly similar in their profiles, but there are some marked differences. But then if you look at people with obesity but no asthma, their cytokine profiles are really quite different. Really, really dramatic. And then likewise, if you compare the people with obesity that have asthma or do not have asthma, again, using these heat maps, you can see that there are really quite interesting differences. So now...
And Michele, one of the things that I didn't see in their patient demographics, but I may have missed it, is did they score whether or not the obese versus the non-obese patients had a diagnosis of obstructive sleep apnea? Because in looking at the microbes that are occurring in the obese patients, many of them look to be dental in origin.
Interesting.
And many of those individuals suffering from obstructive sleep apnea have this tendency where, because of the weight, they effectively, while they're sleeping, obstruct their airway and stop sleeping, and then they aspirate. And that could be introducing some of the microbes into it. While the non-obese, if you look at that heat map one, that's what prompted me to ask this question. And it could be a function of the severity of their sleep apnea that is going on here. But that's just me thinking because I've been brainwashed.
I think that's very reasonable. No, and of course the teeth are going to be intimately associated with the bronchial space.
And there are a large number of folks on CPAP machines, and that would be another good variable to control with whether or not you're using one of the positive airway devices to force air into your airway while you're sleeping, and that's effectively vaporizing the microbes in your sinus cavity and blowing them into your lungs.
Yeah, I'm sorry I don't have the answer to that. I do know that they were looking at, not with people with severe asthma, but with mild to moderate asthma. And whether they naturally excluded those people from this study or not, I couldn't tell you, but I'm sure that that the authors will share that with us in the future. So from the heat maps, they then wanted to look at more details. What are the relationships between particular microbes and particular cytokines in each of these populations? And again, they use analytics that generate really clear, to this naive scientist, plots that you can look at in Figure 4. And again, see that there is a striking difference between people who are obese but don't have asthma and people who are obese and do have asthma. Quite striking difference in the graphic correlations between particular immune mediators and just immune mediators in this Figure 4. They then take it another level and ask, okay, now let's integrate our data sets from the microbe patterns and the cytokine patterns and use what is called a network analysis to generate patterns. So of each of the individuals we've collected and each of the samples, how often are two microbes found together, or is this microbe found with that cytokine, et cetera. So you can imagine with the dozens of different parameters that they looked at, there were a lot of relationships, and to do this by hand would be impossible. But they have the software, the computational tools to generate these clear maps. And what we can see in Figure 5 is that for people that are non-obese and have asthma, there are really only two different network patterns that came out. So they found a particular Prevotella, for example, was associated with particular cytokines, and another one that was more complex that had some different microbes in it. But then if you compare those two networks to what they found for people that were obese and had asthma, now they found 5 different network clusters of particular microbes with particular cytokines. So they're beginning to kind of simplify the patterns that they observed and the interactions between particular microbes and particular cytokines or other inflammation components of the immune system. So this is beginning to clarify a foundation that will allow them to do then hypothesis-driven research to ask whether any of these particular relationships were critical in driving the outcomes of patients with obesity that also have asthma. They then went one more step, and that was to look at not just who's there, but what are those microbes doing? So for this, they again used metagenomic analysis, but they wanted to know what were the metabolic capacities of the microbes that were present in each of the patient populations. So again, that was using metagenomic analysis and sophisticated computational tools. And what they found is that in people that were obese and had asthma, they had microbes that were more frequently had the genomic capacity for fatty acid synthesis and metabolism, glutathione metabolism, lipoic acid metabolism, insulin signaling, and inositol phosphatase metabolism. And that was in contrast to the metabolic capacity of the microbes in people that were not obese but had asthma. In those cases, the pathways more prevalent were associated with porphyrin and chlorophyll metabolism, amino acid degradation, including metabolism of D-arginine and D-ornithine. So again, they're now coming up from this really complex samples of sputum, of blood samples, and of the induced sputum. They're coming up with some clear and reproducible associations that they can now use to develop specific laboratory tests to begin to investigate whether the cause and effect relationship. The authors are very careful and say right now they're just identifying associations. We'll need to do another round of investigation to see which relationships are causal, for example. So, for example, if they can use experimentals of obese animals that have asthma, they can interrogate the impact of particular microbes on the inflammatory response, etc. So ultimately, their goal is to identify host pathways that are distinct in the obese patient that has asthma so that they can target treatments to effectively treat and reduce the airway inflammation of their asthma. So those are the broad approaches that they took, and it is a really lovely demonstration of how we can take patient samples where we understand their clinical profile and then do this really careful work with obtaining biological samples and learn about the microbe and immune interactions, and then begin to use that as a framework to do the next round of studies to ultimately help this patient population.
I mean, I love this paper because I think it's a great example of like a microbiome paper where they sort of have a flood of data, but then they're able to kind of make sense of it by looking at these different associations between different groups and that they've gotten these samples from. I mean, again, this is like goes back to this idea of like a PhD has the ability to take this data and make it into something we can understand to manage it. And it's a really beautiful that way.
Yeah.
You mentioned, we need to, this is an association. We need to do something. What else can we do now to go beyond association, for example?
Well, certainly funding for this type of research would be useful. And also,
that goes without saying.
Yep. Well, they point out a couple ideas in their discussion, some of the things they'd like to do. Investigate the microbial gene expression patterns in people with asthma and obesity and those without. And then look at some of the candidate microbes that they've identified in these associations as a way to implicate particular microbes or particular inflammatory pathways. And they also point out that this first set of data was from a sizable but not huge population of people. So they would want to recruit additional people from their ongoing trials to sharpen their data and push it further.
Right.
So Petra, can you tell us about the first author?
I can. So Ari Kozik, the first author on this paper, now has her own lab in Michigan in MCDB, right? So in the molecular cell biology department. A really great building. And that has the Natural History Museum in it. But so what I like, she told us a little bit about how she got into doing microbiology. And I really appreciated this because her parents must have sensed that she would be interested in science, and they got her a microscope, and her mom showed her how to make slides and look at things in the microscope. And she bought her Petri dishes and agar. So then Dr. Kozik, as an 8-year-old, sampled the bathroom for different kinds of organisms and came up with a whole culture collection. And she says her parents
she always washed her hands after using the bathroom after that little project.
I'm sure. I hope so. But she said her parents were both amused and horrified. So she wanted to do research. I mean, you can certainly get a reaction from your parents by doing that. So she went to college, and she was interested in human behavior, but she was introduced to the microbiome and bioinformatics in an HHMI-funded course that we've mentioned many times on this program. It's the Phage Hunters course, which has gone through several names. When she did it, it was called CURE, but I think now it's C-Phages. And that kind of hooked her on doing research in microbes. And she completed her PhD training on the gut microbiome in mouse models of inflammatory bowel disease, and then went on to postdoc at Michigan in the pulmonary division with the aim of developing expertise in translational research and working with clinical data sets, which is exactly what she's doing here. And she has just started her lab this summer, and I peeked at her website, which is really nice. She has a couple students already, PhD students, and they're building on some of the findings of this paper that we just discussed. And she talks about how the data being collected in her postdoc lab in the Wong lab really kind of led to this study. And I think, this really goes back to these data sets can be mined for lots of different kinds of information. And Dr. Wong used these culture-independent methods first to show that, like Michele mentioned, that the lung has these microbiomes, which was a little bit of a surprise. But then to also separate out asthma, where the symptoms seem pretty similar from person to person, but that probably the cause of that asthma is different. This is adult asthma, not, always children. And I think my favorite sort of thing that when she's talking about this research, she says when she started looking at the microbiome data, there were clear differences associated with obesity. And that doesn't always happen. And so I can imagine that was like a great day in the lab when you could see I see actually clear differences between these samples. And you can see in this paper, they are very clear. And so that's always nice to get clear, clean data like that. I think her advice for young scientists is that I feel that it is the collective responsibility of the scientific community to ensure that we do our best to create and maintain environments where everyone can thrive, and that trainees often end up leading the way forward by asking, sometimes loudly, for what they need. And so she says to junior folks, keep asking, and to the more established folks, please listen and then ask. And then act, sorry.
And Ari has been a real advocate for the community and for the future. She is one of the co-founders with Kishana Taylor and Chelsea Spriggs of the Black Microbiologists Association. This is a 501(c) nonprofit that was formed in 2021, and this was really in response to the June 2020 killing of George Floyd by the police in Minneapolis. Within months, they had formulated a Black in Microbiology Week to really celebrate and build community among Black microbiologists. And they were motivated to do that because in 2018, according to an NSF report, less than 5% of doctoral degrees in microbiology were awarded to Black scientists. And of the tenured professors in life sciences in the U.S., about 2% are Black and only 1% are Black women. So they formed first this Black in Microbiology Week. They had 2,500 registrations from 50 different countries, and they're currently this week having their fourth annual Black in Microbiology Week celebration, which included 6 hours of keynote addresses and sessions on education, research, finding jobs in industry and government on Monday and Tuesday and Wednesday. And they also now have got the Black, this is a component of the Black Microbiologists Association, and their mission is to increase Black microbiology professionals as leaders at institutions, journals, funding agencies, and corporations, to increase science literacy within the public, and also increase interest in the microbial sciences, and also to increase collaborative efforts on the needs and concerns of Black communities across the world. So the Black Microbiologists Association has got a website with a lot of resources. They also have a talent network that they use to build community and help each other advance careers and just create an environment that is welcoming and supportive of our Black colleagues. So Ari is currently the vice president of the Black Microbiologists Association, and as I mentioned, she was one of the co-founders. So she's somebody I admire not only for her ambitious science, but also for her advocacy to try to advance our field and also our community.
And she actually won the 2023 William A. Hinton Award for the advancement of a diverse community of microbiologists from ASM, so American Society for Microbiology. And that's just great that she's been recognized for it. And again,
that's one really of ASM's top awards, isn't it?
It's one of their top awards. She shares it with Kishana Taylor, but they got it together for developing Black in microbiology. And it's been really, I think, very positive.
Yeah.
Didn't we have them on TWiM?
We did. Yeah, one of the earliest. I think it might have been the year that they formed their organization.
If I can find it here.
It was in October, and it was maybe 2021.
It was in the middle of the pandemic, that's for sure.
Yeah.
Well, I'll look it up. I'll put a link in the show notes. We also had, I don't know if it's the same group. There's another group. Is there a Black in virology? I know there's a Black in immunology. We had one of them on TWiM as well. I don't remember. I'll find a link and put it in the show notes.
But for all our listeners, please go to the Black Microbiologists Association website and share it with your colleagues, with your trainees, your students. Help us build this community and support a robust pipeline so that we can address this really horrific disparity in representation in our field.
Here it is, 228, Black in Microbiology with Nanisha Scott and Chelsea Spriggs, two different people from this.
They may have been the officers that year.
Yeah, they're the founders. of Black in Microbiology. Look at that. Wow, there's an alio there on that episode. And all right, we'll put a link to that. I want to read one letter before we leave here. This is from Elijah. Dear TWiM, I love that you stand against climate change and notice every time when you begin to talk about the subject during your weather updates. And so I have a question for you all. Have you ever come across a pattern of emerging disease as related to climate science? I've been curious about this for a while now, and I feel like you are all the right people to ask. I'm a high school student in Michigan, and so I experienced the changing climate through changing winters and rain. But what if, as forests are cut down, livestock populations increase, and wildlife loses natural habitats, that there is potential for disease to emerge from isolated places? On a similar note, are any of you involved or have any colleagues that are involved in climate science? I'm a student at the Battle Creek Area Math and Science Center, and each year we host an annual climate change symposium geared towards other high school students with the goal of encouraging a higher level of understanding on the topic and what can be done about it. I was wondering if you could put in a good word for us. So we will sure do that. Elijah has given us a little summary of what the symposium is and some links to their website and Instagram, YouTube, Twitter, TikTok, etc. So we'll be happy to put those in the show notes. But back to your question. In regard
to your question about climate change impacting infectious disease, I mean, there is clear data on that now. What are the number of cases that we have in Florida of endogenous, not travelers, but endogenous spread of, is it malaria?
Yeah.
Malaria.
Absolutely.
But malaria was endemic here in the 19th century. That's because we eradicated it with DDT and now it's moving back. So, but I think that, the loss of habitat, Lyme disease and a lot of these tick-borne things is definitely a loss of habitat.
And also the populations that harbor those microbes now have a greater range. And so they're not just in, the Texas and Florida, but they're moving our way.
So definitely
Florida has 7 cases of locally acquired malaria, and Texas has 1. P-Vivax. Yeah.
And we're going to get...
I'd also like to point out to Elijah that the organization that Michele used to be president of, the American Academy for Microbiology, actually has a resource called Climate Change and Microbes, a Scientific Portfolio. And you can sign up for a climate change mailing list on their website. So I'll drop that link into the show notes, especially for you, Elijah, so you can learn more about microbes and climate change. And it's something that the American Society for Microbiology is actively pursuing through its members.
I also want to point out that on TWiV 896, we did a paper called Climate Change and Viral Zoonosis Risk. The whole idea there being, as climate changes, animals move because animals always go to where they can find food. And climate change may eliminate foraging for animals. So they go somewhere else and they bring their viruses and bacteria with them and hence can redistribute them. And so that paper describes modeling studies that look at that, and that's another concern. So it's not that you rip down the forest and suddenly things jump out. Certainly the people who are ripping down the forest can acquire infections. We see it happening in the Amazon where they're deforesting all the time. But the real key is during climate change when you have mass migrations of animals, which is already happening. And it's not just mosquitoes, right? It's other kinds of animals that are moving. So a lot of work being done.
There's also plant connections. So plant diseases can be very sensitive, especially when they're stressed by drought. Plants become more sensitive to diseases. So it can really also hurt crops in that way. So that's something else that we don't always talk about, the plant infections.
But maybe, Elijah, a PubMed search with climate change and microbes in the search query, you might pull up a lot of these papers.
Well, that is TWiM number 298. You can find the show notes at microbe.tv/TWiM. And as I said before, if you enjoy our work and you learn something from it, please support us, microbe.tv/contribute. If you have questions or comments, twim@microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you. Good to be with you.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin is at Washington University in St. Louis. That's the full name. Thank you, Petra. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.